Morning, Lily.
Good morning.
You came just at the right time. I was about to start going over recitation 8, but to begin, do you have any specific questions? It's just you and Joseph here, so I don't want to waste your guys' time if you have questions either about recitation 8 or just anything in general.
Yeah, it's on Rec 8, but it's questions 12 and 13.
Okay, that sounds good. I'll start with that then instead of going through everything. So I think let's go to the, you say questions 12 and 13 on the quiz?
Yeah.
Okay, gotcha. So which question did you have specifically? Like almost like how to start it or?
Pretty much everything.
Okay, gotcha. So, all right, let's see a sequence for the coding strand, which makes sense. It's going 5 prime to 3 prime. A mystery gene shown below. Nucleotides are spaced out every 10 bases for legibility. Okay, let's see. Roman numerals correspond to the highlighted sequence directly below. So, after I guess someone is designing primers, they decide to use these sequences. Does that make sense? Like what primers do in PCR and all of that stuff?
A little bit, but the lecture videos really did not help with
Okay,
Mod 5.
gotcha. So primers, similar to almost like DNA replication, they prime for I guess DNA amplification. So when the primer sits on the DNA sequence, that'll recruit the polymerase used in PCR to actually start amplifying the DNA. So it's almost like I don't know, almost like a marker. Like X marks the spot, the polymerase is like trying to find that primer, if that makes sense. I don't know if that made any more sense.
Yeah.
Okay, cool. So they ended up the main thing that you have to do in PCR is obviously you have like a bunch of DNA, but you have to find where exactly you want the PCR product to be. So you like design primers around the DNA that you want to amplify. So, anyways, they ended up finding these primers. Then questions 12 and 13 are asking um, which of the four regions here would the forward and reverse primers bind to? And then the hint is that the forward primers bind to the template strand. So, in other words, this forward primer sequence is going to be complementary to one of these four um, sequences. I don't know if that makes any more sense.
Not really.
Okay. So, well, actually, sorry, I messed that up. The forward primer binds to the template strand meaning it's going to be complementary to the template strand. Up here we have the coding strand. So this primer sequence should be identical to one of these sequences up here because if you have here, let's see if I can annotate on this. If you have your two DNA strands, oh gosh, kind of a straight line. And this one up here is 5 prime to 3 prime. Then that's going to be your coding strand. That's what we have. But the template strand is going to be the strand complementary to the coding strand. It can be 3 prime to 5 prime. So the forward primer is going to bind like, let's see if I could change colors. So the forward primer is going to
I don't see
bind.
any writing on my side.
Oh, you don't?
No.
Hmm. Let's see. Meg or Joseph, can you see like the writing I'm doing kind of like off to the right a little bit? I don't know if you can see it. Lily, it's just like I'm like writing like next to the question a little bit, so that's not showing up at all.
You're still on the beginning slide, the title slide.
Oh, really? Oh, geez. Oh, gotcha. So I started here. I'll try stopping my share screen and then starting again. Did that update it?
Yeah,
Okay, perfect. So, um, yeah, so looking at question 12, which of the highlighted sequences in the coding strand matches the location that the forward primer would kneel to? We're looking at this forward primer sequence. Do you see me highlighting the sequence?
yeah.
Okay, perfect. So this sequence is going to be identical to one of the sequences up here because forward primers bind to the template strand. So what I was drawing here, let's see if it shows up now. So we'll pretend like these are two DNA strands, one going to 5 prime. Can you actually see this drawing now?
Yeah.
Okay, good. So up here you have like the top one is going to be your coding strand, and then the bottom one is your template strand. I'll just name it like C and then T. So your forward primer is going to be, let's see, your forward primer is going to run 5 prime to 3 prime. So it's going to bind to like this template strand. But the sequence itself is going to be the same exact as this sequence up here.
Okay.
So yeah, so that's why when you're looking at um, this forward primer sequence, what you're going to see is that this sequence going 5 prime to 3 prime is going to be the exact same as one of these four up here.
Okay.
Yeah. So I think that'll help you answer question 12. But question 13, the reverse primer is a little weird um, because um, when you're looking at the reverse primer, it's going to be the reverse complement of the coding strand because it's going to bind up here. It's going to mirror the template strand, but from the primer's point of view, this right here is 5 prime, and then this is 3 prime over here. So when I say reverse complement, when you're looking at the actual regions 1 through 4, um, you'll be looking at this sequence, the reverse primer sequence, and then meaning reverse complement, meaning that starting from here, this T, you're going to be looking at these regions and looking at which one is complementary to T. So you would start with either 3 or 4 because the T would be complementary to the A. And then from there, you would look at the A. As you can see, these start AT. And then you would go like trying to figure out which one of those is going to be the reverse
Okay.
complement of this one. Yeah.
Okay. Thank you. That helps.
Yeah. Yeah, of course. Yeah, primer design is really weird just because like PCR overall is kind of a weird concept. So good questions. Do you have any more about the quiz?
But no, but I might just stay muted while you go through
Okay, that sounds good.
the others.
Yeah, so I'll get rid of this drawing. Um, let's see. So I'll stop sharing my screen and then start sharing the slides just to go over a couple things about PCR. Okay, so the overall like review of the basics of DNA, I think I'll skip over that for now, unless either one of you have a question. But um, yeah, so this is the PowerPoint slide with the coding versus template strand that I wanted to make sure to add to this PowerPoint just because I do think it helps with the idea of primer design. Just because if you were to have a primer that binds to the template strand, it's going to be the same sequence as the coding strand. And then with the reverse primer, it'll be the reverse complement of the coding strand. So then just looking at PCR overall, um, biologists were able to almost invent the field of molecular biology because of our understanding of DNA as a whole. So once the structure of DNA was elucidated, we were able to figure out ways to kind of mimic enzymatic activity for like, polymerase and all of the cool enzymes like you've learned about for DNA replication and RNA transcription and use those to like pretty much our advantage. So it's this technique that enables us to amplify a region of DNA of interest. So as I mentioned in the video, cool like applications are maternity paternity testing, forensic investigations, virus testing, and population genetics. So you can actually look at markers um, in a certain population's genome to figure out like specific genetic um, characteristics of certain populations. So looking at the components of a PCR reaction, you obviously need the DNA that will be copied. So this can be an entire genome, but uh, the part of the PCR reaction that actually promotes the specificity of PCR are the primers. So these are the short single-stranded DNA sequences that guide DNA polymerase to actually replicate a specific amplicon or a PCR product. So then, of course, you need the nucleotides to actually build the new strands of DNA. And then this Taq polymerase is a specific DNA polymerase that is still highly efficient at high temperatures. So PCR is usually done at nearly boiling, like a near boiling point, because that allows the DNA, if it's like folded onto itself or still wound, it allows it to unwind, and then the DNA strands also denature, meaning that both strands become separate so that the polymerase can start using those as templates to create the new PCR products. And then usually with reactions like this, you need some sort of buffer that provides optimal like pH conditions and things like that for DNA synthesis and for Taq polymerase to actually work. So once again, here is like one step of PCR or one cycle of PCR. In order to amplify the DNA, you have to have the denaturing of both strands, and then your primers anneal to the DNA, hopefully specifically around the area of interest. And then the extension allows for the synthesis of the new DNA strands. So you're going to end up with two copies from the original one. Also, with this, I like this diagram just because like it simplifies everything. But these primers, they're not going to be like right next to each other on the opposite strands. They're probably going to be like hundreds to thousands of base pairs apart. So you're going to have like one over here and one over here, and then they're going to create like a DNA fragment that's probably hundreds or thousands of base pairs long. So then PCR is really cool because you can amplify DNA at a tremendous rate. So looking at 2 to the n after n cycles, you can end up with a lot of DNA fragments after only a certain amount of time, or like a little bit of time. Um, because each cycle has to go through, and it takes about 5, 6 minutes, depending on how big your DNA fragment is going to be. Um, it can take like hours, but still, it's a great way to actually amplify DNA. So, kind of like I mentioned in the first part about the primers being specific, the primers are going to look like this. So if we have the template DNA up here, with the template being this top strand, so up here on this top strand, it's actually going 3 prime to 5 prime. You would have the forward primer, which is going 5 prime to 3 prime, binding to that top strand, while the reverse primer is binding in the opposite orientation. So then one thing, this is a little abstract. So there's really good videos that look at PCR, actually visualizing it. You visualize PCR products using gel electrophoresis. So, kind of like the name mentions, you have this gel that's made of agarose. Also, sorry if there's background noise. There's a like big truck outside the window. Um, you have like this gel that's made of agarose, which is kind of like a porous gel. So it's almost like Jello, except if it had like a bunch of like little holes and pathways in it. So of course, when you run a PCR, everything is clear. Like you can't see if the PCR actually worked. So as a validation step, people use gel electrophoresis. Or if there's like a forensic investigation, you can use gel electrophoresis to visualize the products of the DNR or DNR DNA to see if they match to any suspect's DNA. So there's this porous gel that you use this like comb almost to create these little wells that you load your DNA samples into. And the DNA sample is mixed with this gel loading buffer that allows the DNA to become more dense and stay within the gel, but also helps us visualize the bands. So it's hooked up to a power supply that runs an electrical current, running negative to positive. And that's important because DNA, because of the phosphate groups in its backbone, is negatively charged. So when you apply the electrical field, all of the DNA is going to run to the positive end. And the bands separate because different sizes of DNA run at different lengths, meaning that bigger pieces of DNA are going to almost run slower because they're bumping into all of these little like agarose, um, kind of like agarose molecules within the gel. So the other important thing is that there's marker DNA, which is almost like a template or yeah, like a template for how big DNA fragment should run on a gel, um, comparative to the marker DNA. So that can run from hundreds to thousands of base pairs as well. And then you use transillumination to actually image it. So then I just included a couple PCR modifications. I'm not sure if there were any questions on this on the quiz. At least I don't remember, but hot start PCR and this final extension time just help amplify the or increase the amplification and specificity of PCR. And it's kind of like a cool like application to molecular biology and how um, we do PCR in the labs. Um, and then one application I thought was super cool. So this was included by TA Aaron because he studies like forensics. And it's really cool how you can use PCR from a crime scene. So if there's DNA left from any biological sample, um, you can do specific PCR and see if it matches with any of these suspects. So then you can collect fresh DNA samples from different suspects and see which bands align to which. So here you would say suspect two might be found guilty because his DNA or their DNA was left at the crime scene. And then this slide I included just because I think it helps visualize what it actually looks like in the lab. So you have this gel with the DNA loaded into it, and it's kind of like submerged in this running buffer or like a gel running buffer. And it just helps supply the electrical current through the gel kind of like evenly. So then after, 30 minutes to an hour even, you then image it in this transilluminator, and then you get a picture kind of like this. So you see like these dark bands on this first like lane here is a molecular marker. So we know the sizes of each of these bands, starting from like, 500 down here, and then going from 1 to 3 kilobases all the way up to 10. So depending on how far down your band runs, you can say that, oh, this product is about 500 base pairs. And this faint one and then this one up here is somewhere between 2 and 3. So that's why the molecular marker is important because if we didn't have it, we just had these bands, we wouldn't know the specific sizes of them. We would just know, oh, this one's shorter. This product is shorter than these up here. So that's the importance of that. And then I hope you guys like the memes because PCR, it can take like sometimes 3 or 4 hours, even like five, depending on how big your amplicon is. So kind of just like hang out in the lab while it's going, maybe do some other stuff. And then PCR is also sometimes inefficient. So If your prime or if your PCR wasn't developed well, like your primers were messed up, one of your reagents went bad, you're going to waste pretty much a whole day's worth of lab, um, because you're not going to actually have PCR amplification. So, um, yeah. Does anyone have any questions about that? Anything I went over? But if not, I'll go back to the quiz and then let's see. Okay. Can everyone see the quiz? All right.
Yeah.
Perfect. Thank you. So, looking at question one, this is just a true false, from straight from the PowerPoint. And then this second question, just going over the matching of the three PCR cycles, which was also included on that diagram that we went over. And then another true or false, looking at longer bands of DNA contain more negatively charged, that should probably say like molecules. So they move further through the agarose gel during electrophoresis, since all of, um, the lanes in the gel receive the same electrical current. Um, this probably won't be the right answer. So it, of course, like I mentioned, has to do with the actual sizes of the DNA molecules. So then, looking at question four, which of the following is an appropriate component for the elongation step of the PCR? You'll notice that only a couple of these are actually required for the elongation step. One of these is required for the denaturation step, which is different and is done at a higher temperature. So um, there's almost like one trick answer in there. And then looking at question five, looking at the SOX10 gene, that's approximately 10 kilobases. How long would it take to amplify the entire gene during one cycle? Well, we know that Taq polymerase synthesizes about 1 kilobase per minute. So you just look at how long it would take for 10 kilobases. So then looking at question six, what type of bond allows for the DNA nucleotides to be built into a chain? Six and seven kind of build on top of each other. Since we know these bonds, acid bound and quectin 6 are phosphodiester bonds, um, you'll know that that's that answer. And then looking down here, these are not phosphodiester bonds, but rather another type of bond. That should be almost like a review question. Looking at question eight, a lab doing gel electrophoresis for like a paternity test. So what you'll do here is, since you have the mother's DNA and the child's DNA, you're going to look at the bands that the child has from PCR amplification and figure out, comparing it to male one or male two, which of these might be the father based on matching of the bands, kind of like how it was with the forensics test, where you're looking at the patterns that the band makes, um, or the pattern that the bands make, and almost comparing it to the suspect's DNA to see which bands align. So then looking at question nine, this is just to familiarize you with like the structural differences between enzymes, and like, different nucleotides as well as proteins in general. Um, so just kind of a question, what reagent from a PCR do you expect this to be, since it is, an enzyme? Um, then going to question 10, you'll just be using 2 to the n to figure out how many copies of double-stranded DNA there are after 12 cycles of PCR. So that leads to question 11. So which is the following statements, or which of the following statements about PCR is correct? Um, the PowerPoint should help you with this, in showing which one is, or which ones are the correct answer. And then we already went over 12 and 13. And then finally, 14, which of the following primers would have the highest melting temperature? I think Dr. Holacek said that you looked at this a little bit in the module concept quizzes or like the learning material. I don't know if that's completely accurate, but we should see that the higher amount of GC content has higher melting temperatures because there are more hydrogen bonds between guanines and cytosines. And then finally, question 15 is kind of a cool application question also where you can look up a PCR product's DNA sequence on this cool website called BLAST that's provided by NCBI, and that allows you to kind of like use the DNA sequence to find where it came from. So it provides specifics on the type of gene it is or DNA sequence, what organism it might have come from. So yeah, it's just a really cool bioinformatics tool that is actually used in pretty much every lab. So if you have any questions about this, I don't know if you tried this. There should have been a link to a video that gave like the steps as well. But yeah, if either of you have any other questions, feel free to ask them. Yeah, of course, Lily.